everybody. Thank you for attending this episode of our education series. My name is Ethan Bourguer. My name is Ethan Bourguer. I'm a member here. We're very interested in understanding more about our cancer, learning more about what drives it and what might help to mitigate it. And so we bring in speakers every month, and we've learned a lot, I think, over the years about what's going on, what are the latest developments in prostate cancer care, and that's, I think, it's been very, very helpful. So tonight, we have a special presenter who's talking to us all the way from Portland, Oregon. He happens to be my brother-in-law, but that's just a coincidence. He has been a cancer researcher for many years at the Providence Hospital in Portland, and they've done a lot of research work on immunotherapy. Now, not necessarily prostate cancer, but they discovered one of the up-and-coming immunotherapy drugs called OX40, which is becoming very prominent now in cancer care. So, since Nick knows a lot about it, and his name is Nick Morris, Dr. Nick Morris, and since he knows a lot about immunotherapy, I thought, hey, would he turn down my request that he present to us? And so, with a little arm twisting, he did agree, and so he's going to give us a presentation about what is immunotherapy, what is the immune system, and what is immunotherapy. So he can give us a nice background. And I think that immunotherapy is going to become more and more important for prostate cancer. Until now, prostate cancer was considered a cold cancer, which the immune system kind of couldn't see. But now there's more and more evidence that at least some people that have prostate cancer also have the kind of cancer that is more visible to the immune system. And I'm sure Nick is going to talk about that. So it's becoming more and more prominent, and Nick is going to tell us that there are now many nice studies out there specifically for prostate cancer, immunotherapy for prostate cancer. So it's coming more and more into the forefront. There's more and more hope that it can provide some real solutions to people who can qualify for this kind of care. So having said that, I think we can turn it over to Nick. And Nick, can you hear me?
Yes, I can.
Great. Okay. So all that having been said, I'll turn it over to you, and please go ahead. And
okay. I was born in LA. I graduated undergraduate at UCLA. Did also graduate school at UCLA. Did a postdoc at the NIH, and also one back at UCLA Medical Center. My first half of my career was in actual structural biology, studying the skeletal system. And I moved over to cancer immunotherapy in 2001 in my 50s. So it is possible to change later in life, although it was painful. But it was exciting to be in a field where you actually can work on something that might end up in a patient rather than doing very interesting but very remote basic research that you don't know whether or not it will ultimately have medical value. So I got a chance to do both in my career, so I'm quite happy with where I am. I think, you know, I'm pretty old. I'm still a year or two younger than Eitan. So, but I have been at this for 19 years, and I'm happy to have the opportunity to discuss the ideas of immunotherapy in cancer, and to point out the potential applications to prostate cancer, which, as Eitan said, has been largely ignored up until recently. But there's a tremendous amount of activity in clinical trial space for this disease, and you know, we always are optimistic, and we're almost always somewhat disappointed, but all progress is welcome, and so I'm going to discuss that. So these are the things I'm going to talk about. So evolution of cancer and its relationship to the immune system. So in the biology of cancer, actually the whole cancer that you have is shaped by your immune system, because there's been this interaction from the very first cancer cell until wherever you are in your current disease. I should point out that I also have prostate cancer. I had prostatectomy and then salvage radiation. And like many of you watching my PSA, which is at 0 at the moment, so I'm in a good spot, but we'll see. Anyway, T cells are the cell I'm going to talk about. There are thousands of cells in the immune system, but T cells do most of that work. And so I'm going to talk about sort of how do they recognize cancer. And then I'm just going to take you through a few of the different categories of immunotherapy, checkpoint blockade, which is probably what you've most heard about, adoptive cell transfer, CAR T cells, and then how to try to make cold tumors hot.
Nick and I stopped for half a second. So can everybody hear okay what you're saying?
Yeah, good.
Yes? Okay, great. Go ahead.
And also don't hesitate to ask questions at any time. I can't see you, so just shout out. So a few sort of definitions and explanations. So some basic biology: genes code for proteins, and proteins are responsible for most of what goes on in a cell. We and I often use the name of the gene and the protein interchangeably, so hopefully that won't be confusing. Proteins can be inside the cell or on the cell surface. And cells talk to each other all the time. And the way they talk to each other, one of the main ways, is when the protein on the surface of one cell binds specifically to a receptor protein on another cell, and that sends a signal to the receiving cell, and it changes what it does. And then a mutation in a gene is a change in the genetic code that leads to a change in its protein. And this can have many different effects on the protein. It can change its structure. It can make it disappear. It's depending on where the mutation and what kind of mutation, you can see ultimately different results. These mutations tend to happen during cell division, when a cell has to make a complete new copy of all its genes. And these mutations are random. So it has been calculated that given all the different cancers, and this is theoretical, that you can 30% of them are due to predisposition or a specific environmental hazard, and 70% are bad luck. Now, people argue about that, but I just throw it out there as something to think about. So some people blame themselves for not taking care of themselves or doing or whatever, but a lot of cancer is bad luck. So how do cancer cells rise? So again, exposure to some insult, chemicals, environmental hazards, smoking, food, work, so on. Some viruses can cause, like HPV, human papillomavirus, I think it is. And chronic inflammation can set off cancer. These insults lead to mutations in genes. The transformation of a normal cell to a cancerous one begins when the mutation occurs in certain kinds of genes that regulate cell division, and these are called driver mutations. So cell division is very tightly controlled by many different genes. That is, in your body, cells need to be quiescent. They're not dividing. They're doing a job, and then other times they need to be dividing, and then go back to not dividing. And there's actually a lot of redundant, overlapping regulations so that the process gets very tightly controlled. So mutations can inactivate genes that prevent cell division, like P53. And those P53 are typically called tumor suppressor genes. That is, they prevent cell division. So if you have a mutation in one of those suppressor genes, then that allows the cell to potentially start dividing. Or it can permanently activate genes that support cell division, c-Myc and BRAF, and there are hundreds. Because this regulation is so redundant, you need 3 or 4 or 5 of these mutations ultimately to transform a cell to a cancerous state. So you need to have these accidents happen. You need to have 3 or 4 of them happen, and they all need to be in the same cell. So again, since these are all random to begin with, it's an unfortunate accident when it happens in one cell and it transforms that cell. This transformation to cancer can be accelerated when mutations are in the DNA repair genes. So mistakes can happen. The enzymes that control replication aren't perfect, although they're awfully good. So mistakes happen during replication. The genome of cells are dividing, but they're very, very rare. Cancer cells divide a lot, so more mistakes can happen. That is, once they lose this control of cell division and they're rapidly dividing over and over again, there's more opportunities for mistakes to happen and more mutations can happen. So in normal cells, lots of these genes are involved in repairing these mistakes under normal conditions, but these repair genes can themselves be mutated, that is, inactivated, so leading to accumulation of even more mutations. So these mutations lead to changes in the cell, and changes in the cell behavior. So what are the results of these mutations? They can kill the cancer cell. Obviously, there are some essential genes even for a cancer cell. So if it mutates too many, it can mutate genes that actually kill itself, or it can mutate genes that enhance its survival and aggressiveness. So we could probably spend the whole talk just looking at this slide, but I'm hoping just to spend a minute on it. So this is sort of the main theory about how your immune system is interacting with your cancer over your lifetime. And it was proposed by Dr. Schreiber, and was published in 2004. And so far, it's held up pretty darn well experimentally that this is, in fact, what's happening. So here is a normal epithelium, these sort of cuboidal cells that sit on an interface between the normal tissue and a space. So these epithelial cells, this is the most frequently mutated cell that leads to cancer. And partly that's because they're exposed to some exterior condition. So this could be cells that line your gut, your mouth, esophagus, your skin, the lumen of the prostate, the bladder. So these cells see a lot of stuff, and they have the propensity to ultimately become cancerous. And they have this very distinct morphology packed together side by side. So what can happen is something, these mutations can occur in a few of these cells, and these should be blue ones here, and they're starting to divide. But your immune system can easily recognize them because these early ones are typically fairly antigenic. Because they've had a mutation, these are now in mutations that are driver genes. They're now different. They're not self, and I'm going to explain the difference between self and non-self in a few minutes. They're more readily attacked by the immune system, and there's a lot of different cells here, macrophages, and so on. I'm only really going to talk about this cell here. I'm going to call it a T cell, and it's actually the main killer cell in the body, and it's the main cell that removes cancer cells. So what typically happens, and happens on and off during your lifetime, is you'll get one of these situations, but the T cells will eliminate it and you'll return to the normal state. And this can, I don't know necessarily how often this happens, but it does happen over time. This next phase is called equilibrium. So a couple of things are happening here. So there's additional mutations. So this cell is different than this cell. The brown cell is different than the blue cell. And the black cell is different than the brown cell or the blue cell because it's accumulated additional mutations. And the T cells are still attacking. And typically there's a strong reaction against the blue cells. The mutation can lead to potentially a cell that loses the mutation that was killing the blue cell, but the immune system can adapt. So whatever new mutation happened here, the T cells can also attack that cell. And and potentially again, the black cell can have a mutation which is even less immunogenic or less visible to the immune system than the brown cell, and potentially still the T cells can adapt to whatever new mutation occurred here, although that interaction is weaker. So you have essentially a Darwinian situation where the tumor cells are changing by mutation, and the T cells are applying selective pressure, killing off the ones that it can attack. And this can go on for a long time because there's basically an equilibrium between the T cell's ability to kill and the tumor cell's ability to escape or to survive. And this is this latent period. So if you smoked heavily in your 20s and then didn't smoke, and then or even if you continue to smoke, you don't get cancer until your 60s. And that's because this kind of process is going on. And then at some point, you reach a tipping point where now the tumor cells overwhelm the immune system and prevent it from keeping it in control. And again, this is based on mutation, and what typically is happening here is that the tumor cells start making molecules that aren't normally made by this cell that suppress the immune system. So immunosuppression in the tumor, based on what the tumor cells are doing, ultimately is one of the main reasons why this escape happens. So the T cells are there, they're heavily suppressed, they can't function, and the tumor just continues to grow. And at that point, you have a clinical disease. All of this is silent and is not detectable typically. You won't see it on scan. It's just happening, and it's not until you get to this point where you have a clinical disease. So what are the modalities of cancer treatment? And you know all of these, surgery, potentially curative if you get it early enough, radiation, same. Chemotherapy targets cell division because cancer cells are dividing rapidly, so there are toxic poisons that you can give that kill dividing cells, but this is hard on normal cells that are dividing, so stem cells and immune cells, etc. And it's as far as I know, in stage 4, never curative. Or I shouldn't say these sweeping statements, but I think in general not curative. You can also, there are also inhibitors of these driver mutations. In prostate cancer, the androgen receptor is in a way a drive. May not be mutated, but the cancer cells are dependent on signaling from androgen, so there's a lot of effort to block that signaling. The same is true of the estrogen receptor and EGF and BRAF and so on. So lots of these driver mutations, and so you can have selective inhibitors of these, but because there are multiple driver mutations happening, you usually just slow the cancer down and it switches over to another, either has a new mutation or it just switches over to cells that better utilize one of these other driver mutations. So there's less side effects because they tend to be fairly specific, but they're also not curative. So immunotherapy is, I think, something that's happening. The first drug was approved in, I think, 2011, which was ipilimumab, and I'll talk a little bit about that. Checkpoint inhibitor. But in immunotherapy, the immune system engages the cancer and kills it. And this can be highly specific, but all autoimmune effects can also rise, and I'll try to explain a little bit about that. So in general, the only approach that we know that I'm aware of that can provide a cure in stage 4 metastatic disease.
Hey Nick, can I just suggest you can just slow down a little bit because it's, I don't know about
sure.
Here, but I'm having a little bit of a hard time picking up all your words. So
okay.
Okay, thank you.
I'll speak more slowly.
All right, thank you.
So one way to look at this is that with chemo and targeted therapy, you can generally get delays. So the patients all eventually die, but they can benefit in an intermediate way, so they get more good years. In immunotherapy, we tend to look at it that for some patients there's no benefit. For other patients, there's a little bit of a delay. But there's some patients who are ultimately cured. That is, their disease never comes back. And our goal, of course, is to improve that response so that we can get a higher percentage. This is aspirational at this point. We'd love to see that, but that's for sure never going to happen. But, you know, we're all working on this ceiling at the moment. And a lot of these immunotherapies that I'm going to talk about are trying to sort of push this response rate.
And again, so do you all understand these graphs? It's called the Kaplan-Meier curve. It's so if you look at one of this. So this is the percent of people who are still alive. So 50% is over here, and so you can see, or let's see over here. So 50% of people on this kind of therapy can only survive for less than a year, but 50% on this kind of therapy can survive for over a year. So you want to sort of, the more this curve extends to the right, the better. And that's what Nick is trying to explain here.
The colors aren't labeled, but I'm assuming the red curve is no therapy at all. It's the same on both graphs. The blue curve shows how many more years you would live with that particular therapy.
That's right. He's comparing different therapies.
Why is the one dash after 1 year in the targeted therapy and chemotherapy? And in addition over here, what's the green and black one stand for? Future developments?
Yes. So the blue line is where we are now. So what we hope is to push this up. And I mean, it depends on whether you're talking about complete or partial responses, because we're pretty good at the 2 to 3 years. But what will happen is that this will just keep on going indefinitely. These patients will ultimately, some of them will come back down to this level if you can carry this out. We shouldn't get too hung up on this curve, and I obviously did a poor job of explaining it, but this basically is a way to measure what impact the treatment is having on the survival of the patients receiving that treatment. And the difference is what we used to call the tail on the curve. So almost all other therapies look like this, where they eventually get to zero. Immunotherapy, yeah, a lot of people aren't benefiting or only benefiting a little, but there's this plateau. These patients just keep on living. And that you don't see with these other therapies. And the goal is to get more people to live along, die of something else, which is how I put it. And but also we're seeing more results where this curve is being pushed to the right, where there's better survival even if it's not a cure. And I'll talk about that in a moment.
The other thing that you didn't point out here is that, from what I've heard, but I'm not, I haven't heard that much, immunotherapy does seem to have different efficacy depending on the type of cancer. From what I've heard, prostate cancer doesn't respond too well to it yet. Brain tumors may respond to it, overwhelmingly well.
Yeah,
this graph here, is this just the generalization of it, or is it specific to one type of cancer?
This is a generalization, and I wouldn't yes, prostate cancer is one that doesn't respond particularly well. I'm going to show you the results of a clinical trial in checkpoint inhibitors in prostate cancer in a little bit. But these are just idealized curves to make a point. Yes, checkpoint or immunotherapy works better in some cancers than another, and that mostly depends on how many mutations cancer cells have, because those are the targets for the immune system. And I'm going to talk about that in a second. So how do T cells recognize tumor cells? So, tumor cells is only a few changes away from a normal cell, and we know that the immune system tends not to recognize us, and we only see that in autoimmune disease, but mostly your T cells going through your body, if they see a normal cell, nothing happens. So how does it distinguish a tumor cell? So what is the protein? So I've represented a protein as a string of beads. They're made up of a linear array of 20 different amino acids. These are the building blocks of proteins. So I've just represented those 20 by different shapes with different colors. It doesn't matter, it's just a representation. But this linear string of beads is made, and then it folds into some sort of three-dimensional shape, and it's space-filling, so it's spherical or elongated. Each gene codes for a unique sequence of amino acid that can vary in size from less than 100 to many thousands. So proteins can be small or quite large. And again, these amino acids spontaneously fold into a shape that is determined by this sequence. So how do T cells recognize the tumor cells? Again, so what happens during the synthesis, when these proteins are made, there's machinery that basically reading the genetic code just adds one at a time until it reaches the end here. But during that process, a portion of these proteins are chopped up into pieces before it folds. And these pieces are on average about 9 amino acids long. And we call these pieces of protein peptides. And it seems inefficient. Why would you waste all the energy of making proteins and then chopping them up? But there's a reason for it. So what happens in the cell is that these fragments of protein, these 9 amino acid fragments, they get loaded onto these protein. This is not one. This is a protein, but it's actually a complex of a couple of proteins. This blue thing I made, and there's a groove in this structure, like it's like a hot dog, and this peptide sits in this groove. There's one fitting here and one fitting here, and so but not every peptide fits. There are certain rules. It's basically a shape dependent, and there's some chemical interactions. So you have to have certain kinds of amino acids at certain positions to make it actually fit. So in this case, for example, 2 peptides fit, and all the rest of these don't fit, and they're ignored. They don't go into the system. Now here I've just turned this 90 degrees because this is too hard to draw over and over again, or to represent. So I just turned it end on, so you're just seeing the green box. But this is the same as this. Look end on. And then these complexes, this MHC molecule.
So,
pardon?
can I just stop you there? So can you just tell us again what does MHC stand for?
Let's see. I think it's multiple histocompatibility.
Right. It's right.
So this is well.
Histocompatibility complex, right? Multiple.
Yeah.
Right. Okay.
Or is it major histocompatibility complex?
Major.
MHC.
Major histocompatibility complex. Okay.
Right. So these complexes made in the cell are then transported to the cell surface. So this part is sticking outside the cell. And while I've drawn just this one complex, there are in fact there are many other proteins involved in this overall process, including the chopping and the moving from one cell compartment to another cell compartment to ultimately getting it out. But the end product is this MHC molecule with this piece of protein sitting on in it and then moved to the outside. So all the cells in your body have a representation of every protein being made in that cell sitting on the outside of the cell. Does that make sense?
It does, but that's really an amazing thought, right? That
yes,
there's all these MHC molecules stuck on the cell surface. That's an amazing
right.
thought. Okay.
Yeah, because that's because the immune system can't look inside the cell to see if anything's wrong. It can only see the outside.
Okay, so this is the way the cell identifies itself to the host.
Correct.
That it's what it is. It's kind of like its driver's license or its identification card, whatever you want to call it. Huh?
Right. It's a way of distinguishing self and non-self.
Okay.
So if all the proteins are normal, then it's self. If there's something abnormal happening inside the cell, a piece of that will load in here, go to the surface, and the immune system will say, this isn't normal. So this is what a normal cell looks like. So this is the nucleus where the DNA is, the genetic information. You have all your MHC with all these different peptides on the surface. And then you have a T cell. The T cell has a special protein called a T cell receptor, and I'll call it TCR. It's also actually a cluster of proteins, but it's a single structure. So we'll just call it TCR. And this cell is going to look at this cell and try to decide if there's any problem going on. And this is sort of just looking at that individual interaction. So the T cell receptor, this TCR, is looking at this peptide, and basically it's again lock and key. It's surface against surface. Does it fit? And there's part of the interaction is between the TCR and the MHC, and part of the interaction is between the peptide that's in the MHC and this part of the TCR. In this case, this is a square, this is a triangle, this doesn't recognize, the TCR does not recognize this MHC, right? So it's going to ignore this cell. And now every T cell makes only one kind of TCR. So this T cell has many of these TCR receptors on its surface, but they're all identical. So this T cell has a single specificity for whatever foreign thing it can find.
So Nick, can I stop you again there? So are you saying that, okay, you say there are hundreds of these MHCs on the surface of every cell, so
Thousands.
There must be hundreds of different TCRs?
There are anywhere from 25 to 100 million different TCRs.
Oh, wow. Okay.
Theoretically, based on how they're generated, the theoretical number is 10 to the 18th.
Jeez.
But only a portion of those actually end up in the TCR. But so that's how you generate the diversity you need to recognize all the different structures that can ultimately be targets, right? Bacteria, viruses, whatever, cancer, mutations, and so on. So T cells can't see normal MHC peptide complexes. So this happens by, I'm not really going to talk about it, but basically when these T cells are made in the thymus, any T cell that has a receptor that recognizes a normal peptide, that cell is killed, most of them. Those that aren't killed are strongly repressed, so they're still circulating in your body, but they've been profoundly switched off, and they don't do anything, because obviously we don't want autoimmunity. The autoimmunity happens when there's a defect in the immune system, and those are allowed to turn on. But we won't go into that now.
So wait,
so what happens? Oh, go ahead.
Stop you there, because I think this is really important. So what you're saying is that, okay, so these T cells, they circulate around, they lock into a normal cell's MHC, and if it recognizes, in other words, if the T cell sees the self-peptide, in other words, the lock, the key and the lock interact properly, then that T cell is basically eliminated because it's not useful, because it's
well, actually, no, it doesn't happen. I mean, this happens in the thymus, not in the rest of the body. So it's a little bit of complex biology that's hard to get into. But the way to look at it is that these T cells all evolved in your thymus, which is a gland above your heart in your chest. And all immature T cells go there, and that's when their TCRs are developed by recombination in the genes, and it's a very complex process. So all these different TCRs are generated, one kind per cell, and then they're selected. And so any TCRs that are in the thymus that recognize any of the normal proteins in the body, they're eliminated. And I'll say this, but I hope it doesn't confuse you, but there are special cells in the thymus that have no regulation of what they express. So they're actually expressing a little of everything. So they express heart-specific proteins, they express brain-specific proteins in small amounts and chop them up and present them on their surface. So as these T cells are getting educated, if there's a strong interaction between a TCR and a self-peptide, that T cell is eliminated. Some get through, especially if the binding isn't too tight, and they're suppressed elsewhere in the body, or they're made tolerant, so they won't react to a normal peptide MHC complex.
Okay, and the whole point of this is that we don't want to overstimulate the immune system. We want this is a regulation, this is kind of regulation mechanism.
Right, because you don't want it to attack yourself. You don't want it to attack self, right? You don't want it to attack your own cells.
All right, so that's the point of it. All right.
Yeah. So then what happens with tumor cells? Well, tumor mutation is when typically one of these, there's many different kinds of mutations, but the most common is where the wrong amino acid is put into one of these spots. So I made it a black, but it's a normal amino acid. It's just the wrong one for that position in the protein. So this mutant protein gets chopped up. Now the MHC is carrying a mutant peptide, and presented this way. The same rules apply. Not every mutant peptide will fit. Therefore, the more mutations, the greater the chance that one will fit into the MHC protein. That's why in immunotherapy, the more mutations you have, the more higher likelihood that there's an existing immune response against those mutant peptides. So if you look at it again, you're looking at one TCR interaction. So now this fits tightly. It's a selection process. All the different T cells with the different TCRs are engaging these tumor cells, and the one cell that fits, right, triggers a response. The strong and the affinity, the tightness of this binding, the goodness of the fit, the better it is, the more activated the T cell gets. And when activated, the T cells can divide every 8 hours, so one cell can make 2 million cells in a week. Typically, there's usually pools, so this TCR, this specific T cell, with this specificity, there won't be one of them. There'll maybe be 10 or 20 or 100 total, but they quickly expand to huge numbers. And another thing that yes, question?
Okay. Repeat that. I think it wasn't that clear what you just said.
Pardon?
Okay. So when you have this tight fit, then you say that energizes the T cell to divide very rapidly.
Yes.
Okay.
And the other thing it does is it becomes more cytotoxic. It turns on a set of genes that allow it to make more and more of the kinds of molecules it uses to kill. So, I'm sorry. So these molecules are spewed out of the T cell and they directly attack the tumor cell. In fact, it basically punches holes in the tumor cell and injects a whole series of enzymes and other things that destroy the tumor cell.
Is that called lysis? Is that what lysis is?
It leads to lysis. basically it's not the way a virus does it, but it basically punches a hole and just destroys the inside of the cell. And those cells can be lysed or they'll be gobbled up by macrophages or whatever. But in the end, the tumor cells are killed. So the signals cause a lot of cell division, plus increase in the number of the amount and kinds of these proteins that are used to kill the target cell. So what happens if the MHC itself is mutated in the tumor cell? So this is a gene that's being made in the tumor cell. Since mutations are random, there can be mutations in this or any of the genes products that are involved in chopping up these peptides and loading them onto the MHC molecule, transporting them around the cell. So obviously, if there's no MHC peptide showing a mutated protein, you will not get a response to this tumor cell. And if you look at failures of immunotherapy, a large proportion of them arise by mutations in one of those genes involved in making the MHC peptide complex appear on the surface of the cell. So again, here is a representation. So here's this cell. T cell has only one TCR receptor. It happens to recognize this peptide. It's ignoring all these others. You have about 500 billion T cells in your body. And there's about 25 million different T cell receptors. Although again, each cell is only making one kind of TCR. And there may be only 10 to 100 cells that recognize this particular mutation, so you need to expand, and that's what I mentioned before. But when activated, these T cells are quite toxic, so they have to be tightly controlled. Now remember, this mutant peptide is coming from a tumor cell, but this is also how it sees infections. This could be a fragment of a viral protein. This could be a fragment of a bacterial protein. And when that happens, those kinds of pathogens send very strong danger signals to the immune system, so they get even extra activation. So in cancer, cancer's immune system is working without those danger signals, so it's not quite as potent as a response as you would get if you had a bacterial infection or a viral infection in the same cell. But nevertheless, the immune system does see and eliminate these cancer cells most of the time. So how is this control exerted so that you can, So again, once this happens, you get an attack on the cell. So you need to control the T cell responses. So they need to be quiet while moving to the right place in the body. They need to be highly active at the site of trouble, whether it's an infection or cancer. They need to be deactivated when the infected or cancer cells are gone. And you need to promote healing. And I mentioned TGF-beta here because I think this is a huge it's a growth factor that helps cells repair. And I mentioned it because I think, we're working on it, so it's important to me. And it's a very negative factor in controlling T cell responses. So if you have this infection or cancer and you have the tissue is like a wound and it needs to heal, TGF-beta comes along. It's a hormone or growth factor. It stimulates the normal cells to repair the damage, and at the same time, it's helping to turn off the immune response. So you need to quiet the T cells, and in the end, you need to kill them and make them go away because you need to make room for other immune responses, like to get the flu or something. You can't have trillions of flu T cells and keep them, and then to have another infection. You need to get rid of those because you only want them there when you need them, and then you need them to go away.
So Nick, if I can understand this, it's kind of a very dynamic process.
Yes, very.
Where the body is constantly, you've got these feedback loops, a constant
Yes.
tell the body how to regulate the regulate this immune response. Okay.
Yes, yes, you get these huge expansions and contractions of the immune cells, the T cells, and because you don't want them hanging around, because then you only have so much space, and pretty soon you won't be able to respond anymore because there's no place to put all the T cells. So, and there's I'll mention it only that there's something called memory. So while you're killing off most of the cells, there's always a reserve pool that are called memory T cells. That's what you get when you vaccinate. So those memory cells, they respond much quicker to the, so let's say an infection, if you get the flu once, you get a good response. If you get the same flu again, you have memory cells, and they can get to that new infection faster, so that it becomes asymptomatic, or you get a very mild disease. So not all the cells go away. It hangs on to a small population of those cells that were affected in that response.
Amazing.
So here is the way that the immune system controls the activity of the T cell. So here are a set of proteins on the surface of other immune, these are sort of accessory cells that help the immune system act. And this is the receptors on the T cell. So again, these are all proteins sitting on the surface. These are other proteins. So when this protein binds to this protein, it sends a signal to the T cell. When this protein binds to this protein, it's sending a signal. There's a mixture of stop signals and go signals. So typically,
Wait, can I again? So what are this stuff on the left? This is one immune system cell talking to another immune system cell, or what are we looking at?
Well, this is a T cell.
That's
And these
and on the left is another kind of immune system cell, like a
yes. They're specialized cells and more generalized cells. They're all involved in helping these T cells get started and helping to shut them down at the end. So, for example, if you get a bacterial infection, you get a very strong expression of these start signals, these go signals, and those drive a much better expansion and differentiation to a killer phenotype, to a stage where the cell can really kill. They can still get there without the danger signals, but it happens much better if there are danger signals present to upregulate these go proteins, these go receptors, to activate them. And then you have these stop when you're done with the infection or whatever. You want to turn off these T cells. These other cells will express stop signals and turn this T cell off, and then eventually they die. Now, again, go signals help start the T cell activation. Stop signals, they come from these accessory immune cells, and these are often called checkpoint inhibitors, these stop signals. It's like checkpoints in the city, in an army, where they're controlling traffic, whatever you call that. But what's interesting is that the tumor cells, by random mutation, can start to express these stop signals themselves. So, for example, PD-L1 is a primary protein that binds PD-1 and stops the T cells. Activated T cells express PD-1. So when PD-L1 comes and binds it, it shuts down this T cell. If the tumor cell is expressing PD-L1, the T cell comes along, sees the tumor, but is engaged by this PD-L1 protein and gets shut down. So it never gets a chance to do its killing. And so that these have become the focus of immunotherapy. So there's PD-1 and there's CTLA-4, so blocking these two proteins. This was the first one to come to market and this was the second one to be approved. In a chronic situation like a tumor, all those during that equilibrium phase and on into the escape phase, these stop signals accumulate on the T cells and on the accessory cells, and also on the tumor makes more and more of them by random mutation. So you end up with a situation, again, I'm pointing out TGF-beta here, that T cells have a receptor for this growth factor. It's a stop signal for T cells. And a lot of tumors express this, and what I've been observing recently is actually prostate cancer is actually very high in that TGF-beta protein. So I think for immunotherapy of cancer, until they come up with a co-therapy that includes an inhibitor of this signal, I think it's going to be hard to make it as good as it could be. That's my viewpoint. I don't have data to show that yet. But we think that in prostate specifically, and in lots of other tumors, TGF-beta is one of these major checkpoints that hasn't been. There are people developing drugs for this. There's none approved yet, but there are many in clinical trial.
So this is a ligand that the tumor cell itself expresses.
It doesn't. Yes, it secretes it. So the tumor cells make TGF-beta, this protein. It's not on the surface. They just secrete it into the space. But obviously, it can float over and then hit the receptor. So it's just a little bit indirect. It's not a direct from one cell to cell. It's the cell makes it. It floats around and comes over here and then sees the receptor and then sends the signal. And it's also, especially in bone metastases, which are prevalent in prostate cancer, bone has a lot of TGF-beta in it naturally, even not made by the tumor cell. And so again, TGF-beta is going to be an important target in that setting. So there are other factors that make it difficult for the immune system. There's abnormal blood vessels, so T cells getting in and out. The pH is very low, which also can impact cells, and the oxygen tension is very low. So there's less oxygen in the tumor environment. So all these things together make it openly shut down the immune system and allow the cancer to grow. So let me take you through some of these modalities. So here is checkpoint immunotherapy. The goal is to reactivate a suppressed T cell in its place in the tumor. So patients are infused with an antibody that binds to either the ligand or the receptor to block the inhibitory signal. So for example, here's PD-L1, here's PD-1, sending a stop signal. You can give an anti-PD-L1 antibody, and I don't know how familiar people are with antibodies, but they are another different part of the immune system, but they can be used to make these proteins which bind very specifically to a single target. And I think we can't go into that now because there just isn't time, but let's leave it that you can make antibodies that bind to PD-L1, or you can bind to PD-1. In either case, you'll prevent this interaction. So this leads to activation of the T cells that reside in the tumor and the renewal of tumor destruction. It tends to work well and is approved in tumors with high mutational load. Again, remember, the more mutations, the more mutant peptides on the surface, the more different T cells will recognize the tumor and attack. And so it's approved in things like melanoma, lung, bladder. There's 9 total tissues, but not in prostate cancer, but that's changing. The overall response rates in these optimal situations, melanoma is the one that seems to respond best. So the overall response rate, stable disease, partial response, complete response in melanoma is about 55%. The true complete response rate, CR, that is complete responses that lead to cure, is still only about 10%. So, for example, in metastatic melanoma, without checkpoint immunotherapy, or if you're at the end of standard of care, your life expectancy is 6 months. With checkpoint therapy, 80% of patients are alive in 1 year, 70% are alive in 2 years, 50% are alive in 3 years, but at this point, only 10% are ultimately alive 20 years later that are cured. So you can see this as shifting that curve to the right, that survival curve. So people are getting 1, 2, 3, 4, 5-year benefits without cures. So it's becoming sort of a chronic condition, but it's not the cure rate we had hoped for. There's some claim that if you combine the 2 checkpoints I talked about, PD-1 and CTLA-4, this is 20%, not 10%. But I talked to my colleagues here, the oncologists, they don't believe it yet. They say there's not really enough data. But I think it suggests, they do agree that the dual therapy is better, but it comes at a high price, which we'll get into in a minute. So this is comes from a classic paper that correlates this tumor mutation with the different kinds of cancer. So on the left is the frequency of mutations in the cancer. Each of these dots is a patient. Each of these is a different kind of cancer. So you can see it's a log scale. I guess some of you are engineers, so you know what that is. Even with a log scale, you can see there's a slope to this line. So this represents thousands to tens of thousands of mutations in the tumor, totally. And this is, 10 or 20 around here. So melanoma is here. Prostate is here, halfway in between. So if you look at who's getting treated, it's these guys. So these cancers have the highest mutational burden, and that's where the companies that are developing these drugs go first, because in order to try these other cancers, you need to get it approved here first. Then you can start working because you can then start selling it and make money that allows you to then go look in these other places. So there are a lot of drugs approved. And again, the number of mutations matters, because the more mutations, the greater chance of having the right mutation. That is, the peptide fits into the MHC and recognized by the TCR.
Could I ask, I don't have my glasses on, the type is a little bit small, but the 6th one from the right, does it say osteophagus or osteophagus?
Yes.
6, 1, 2, 3, 4. Oh, I don't even know what that one is.
I'm sorry?
I've never heard of it.
He doesn't know what it is.
Oh, but it could be esophagus, although I've never heard spelled that.
I would commonly call it bone cancer?
No, that would
That would be
osteosarcoma is bone cancer. This is esophagus. This is where you swallow.
Oh, esophagus. This must be an
They're calling it OE. This must have been an Englishman that wrote this, because we don't spell it with an O.
And am I mistaken, but prostate cancer does not appear to show up on the list at all?
It does. It's right.
It's right there.
Oh, okay.
So it's relatively, compared to over here, it's 10 times less mutations, right, on the median number of mutations.
Right, it's about a log level.
Smaller.
Right. Okay. So yeah, I should have done this before. Here's prostate.